Okay. First of all, thanks for giving me a second chance. So, well, I'm Damiano Piovesan from the Biomedical Sciences Department at University of Padua in Italy. I'm going to talk about the disorder ontology assessment, which is a new challenge of the CAFA4 initiative. Well, the challenge is pretty much the same as the gene ontology prediction challenge. The only difference is the annotation growth phase. For the disorder ontology, we provide annotation from Disprot, and we annotate intrinsically disordered proteins. Intrinsically disordered proteins are a specific class of proteins which contains intrinsically disordered regions, which are regions which don't have a fixed three-dimensional structure. They also are highly regulated. They can be easily accessed by post-translational modification enzymes. They are usually involved in signaling, transcription, and translation processes. And they do so because they are able to perform a special type of binding. They have high specificity. They have higher surface of interaction because they are linear compared to a globular domain. They have low affinity because there is an entropic cost of adopting or adapting to the interacting surface, and they provide a very high versatility. An example is P53 in his C and N terminal regions, which are able to bind many different partner proteins. So Disprot is a database of manually curated intrinsically disordered regions, and all the annotation comes from the literature. Disprot is not a novel database. It was created by Kit Dunker and was first published in 2005 in Bioinformatics. Since 2017, it is hosted at the University of Padua, and it is a community curation effort. So there is about up to 40, 50 people every time, at every round, coming from community built on the NGPNet cost action 4 years ago, and which is now also supported by the Elixir IDP community, which provides annotation for new targets. In the case of CAFA, we restricted the curators to only 3 expert curators, and they were enrolled, let's say, to generate disorder function annotation for the CAFA targets. So I think I have to say something about the ontology. The ontology is new, but it's based on a control vocabulary, which was first defined by Kit Dunker, Peter Tompa, and Vladimir Uversky. They basically provide a classification of different type of disorder functions. And Disprot, since the release in November 2019, has formalized this control vocabulary in the OBO format, which is typical for the ontologies, and try to give more structure to the terms and their relationships. At the moment, it contains 5 different namespaces or branches, which are quite independent each other. And namely, they are structure state, structure transition, interaction partner, disorder function, and detection method. Detection method terms are used only as evidence codes, so they are used to qualify the quality of an annotation or the provenance of an annotation. In CAFA, we decide to only consider disorder function terms, which are more meaningful, and I will explain why. Disparate annotations are provided at the region level, so you see that each functional region corresponds to an article, and in particular, each functional region is characterized by a term, which describes its function, a position, start and position of the region, and the evidence code, which in this case, in all cases, it's an experimental biochemical method. For CAFA, we only consider the functional terms, as the position is too difficult to predict, in our opinion. And the evidence code is not relevant, as all the piece of evidence comes from the literature and comes from experiments. So the ontology is small. For the disorder function, you have 48 children terms, and the depth is just about 3 levels. It's also not DAG, it is a tree. And you see here the children of, for example, the structural state branch, which contains only ordered and disordered terms. And 100% of the proteins in this spot contain a disordered region. So it makes no sense to try to evaluate if a predictor is able to say this protein is disordered. This is true by default. For order, this is different. And the order term is very rare. It's used 5 times in the entire database, and it's used only to highlight that some specific subregions, for example, are ordered inside these other regions. Lateral transition also is small. Interaction partner can be perfectly mapped to the gene ontology terms. So it may be more appropriate to be evaluated at the gene ontology challenge. And also they are not quite curated at the moment. And so we concentrated on this disorder function. So this other function contains 7 different children, and most of them are related to binding events, so to biological process related to the binding events. With the exception is the entropic chain. I will show right now some example. The flexible linker spacer term is a lift term, and for example, in this case, it shows that a region is used to allow movement of domains or regulate the distance between 2 domains. In this prot, this is the identifier. This is not yet public, and it's kept private for the moment. And what I wanted to say is that for each annotation, we also provide the snippet coming from the corresponding article, which the curators decided to report in order to describe where the annotation comes from. Another example is the molecular recognition effector. In this case, the term is not a lift term. And this term explains a function which is related to the modification or modulation of the activity of a partner molecule, or modify the activity of another part within the same protein through competitive interaction or through allostatic modulation. You see that these kind of terms are very high level, very abstract somehow. And curators in this plot are also allowed to annotate terms which are not leaves. Okay, this is another example. In this case, you have a child of the molecular recognition assembler, tethering. Tethering is a lift term. In this case, you have a disorder region that serves to tether a weak affinity binding region to increase its local concentration. Again, it's one term which describes a complex phenomenon. A sibling term, targeting, maybe this is more easy to understand. This is correlated often with linear motifs, which are well known to contribute to subcellular location. What the data set? So our data set, our curators were able to annotate 263 new proteins, CAFA proteins, and compared to the training set, so the proteins which were already available in 2018, we have a data set which is a ground truth which is about the same size, both in terms of proteins, in terms of terms, of course, and in terms of leaf terms. What's interesting also is that only 6 out of these 263 proteins were already annotated with a functional term in 2019. So for the evaluation, we only considered the no knowledge mode. So the almost all proteins are new targets. So in terms of organisms, the majority of targets comes from Homo sapiens, from human, and all the rest, with the exception of less than 20 proteins, comes from eukaryotes. Unfortunately, viruses are not represented. Maybe the curators didn't pick up those proteins, or maybe they were available from the CAFA organizer. I didn't check. I evaluated sequence identity of the ground truth versus the training set. There is a number of proteins with 0% identity, and about 60% of the proteins has less than 40% identity. Maybe this is not super relevant for predicting disorder, but at least for specific binding regions, the identity or the homology is important. And also you may exploit the fact that flanking structure domains of these other regions are conserved. So you may, for example, if there is a linker and where the function is to separate two domains, well, maybe looking at those domains, it also helps in predicting the linker position or the linker annotation. Well, the ontology is small, so you can visualize it in the distribution of each term in the ground truth in the training set. What you can notice is that flexible linker spacer is the most frequent term, followed by subset of terms which are particularly abundant or are overrepresented. If you look in detail at these terms, they are regulation of phosphorylation, which is child of molecular recognition display site. You have molecular recognition effector, and you have a class. You have flexible linker, of course, and you have a cluster of terms which are molecular recognition assembler and children of this term. So somehow there are 4 branches which are highly represented, with maybe 3, 4 of them which are even more represented. This is the distribution when you add ancestor. So in the disorder ontology, you can apply the same you can do with gene ontology. So you can propagate an annotation up to the root. And see again that if you count the number of entropic chain flexible linker spacer, they account for about 1/3 of total terms. Whereas molecular recognition, effector, assembler, and display site account for the other 2/3 of the terms. Maybe I haven't explained that, but some of the terms are only available in the ground truth because they were introduced to refactor the ontology very recently. So they were not available in the training set. So probably they are very difficult to predict. So the results. The assessment has been performed using the CAFA assessment tool from the repository developed by the CAFA organizers. And we just modified that tool in order to work with the disorder ontology. We generate the ground truth in the proper format, and that's it. So we didn't reverse engineer the software or modify it. We consider both the full and partial evaluation, where the partial means the recall is normalized over the predicted terms and not over the entire data set. And as I said before, we only evaluate the no knowledge or no previous knowledge mode, as all the targets are new targets. And we provide precision recall curves. Okay, this is the first slide. I haven't said anything about the baselines. We implement 2 baselines, one naive and one blast. They are identical to the one provided by CAFA. The naive just transfer terms to all the same set, all the terms to all targets based on their frequency in the training set. Blast instead assign a confidence based on the sequence identity of target versus the training set. So what you can see is that the best method, which was just presented before my talk, is a slightly better F-score compared to the baseline. And also there are a couple of terms which are very low coverage. So they predicted like 10% and 3% of the total targets. So apparently the disorder prediction, the function of the disorder function prediction is a very difficult task. And the same happens when you evaluate the partial, you consider partial evaluation. So again, the F-score is quite low. So what we inspected at this stage was which are the terms predicted at F-max. So when you consider the threshold obtained at F, when the F-score is maximum, and you keep all terms with a score above that threshold, you can see the confusion matrix, the true positive, false positive, and false negative for all the terms in the ontology. And for the naive method, you see that it just predicts flexible linker spacer and get almost the best F-score. So the idea was to try to remove this flexible linker spacer and see what happened. And the idea at the basis is this, to simulate what, or to think about what happened in the first two editions of CAFA, in which the protein binding term, which was associated to almost all proteins in Swiss prot, affected consistently the ranking and the evaluation. So this is the performance removing the linker term. So we consider in this case about 200 proteins, but still the performance is, the best method is slightly better than the naive, but still the performance is not that that good. So it is and the same happens also for the partial evaluation, and this method here, team 3, only predicts 7% of the protein, so it is not very meaningful for the evaluation. So just for reference, this is the behavior of the naive, the moving the linker space, the linker term. So you have the positive predictions, which concentrate on the molecular recognition effector and the molecular recognition assembler. But still, you have a lot of false positive, of course, by definition. This was last slide about the evaluation. So my opinion is that the disorder ontology is difficult to predict. The annotation of disorder are difficult to predict, either because terms are somehow way too abstract, or they escape the classical structure function paradigm. Or the other reason could be that the training set is very small. So maybe as we just recently concentrated in increasing the number of functional terms inside this prot, it could be solved in the closed feature.
Damiano, sorry. Oh, good. You're on your acknowledgement.
Sorry. I was asked to acknowledge the organizer, Silvio Tosato, who collaborated and helped me in the evaluation, Petra and Hido, and in particular the curators Federica Quaglia from the University of Padua, Lucia Chames from Universidad Nacional de San Martin, and Rita Pangsa from the Hungarian Academy of Sciences. With that, thanks for your attention.